Hello, everybody, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Workflow Solutions for High-Speed Glycan Analysis, from glycan prep to separation and mass spec analysis. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. The analysis and assessment of glycosylation in proteins is one of the most critical steps in the development of biological drugs. Efficacy, safety, bioactivity, half-life, and immunogenicity are all dependent on the glycosylation pattern. Today's webinar has been designed to show you how to establish and carry out an efficient and economic workflow for successful N-glycan analysis. Our panelists are going to focus on the key steps of sample prep and separation optimization. Let's meet our expert team. Dr. Oscar Potter is a research scientist at Agilent Laboratories. He will look at the use of a specific approach to the analysis of therapeutic protein and glycans. James Marticella, an application scientist in the chemistries division of Agilent Technologies, is going to talk about glycan sample preparation. Dr. Sonia Schneider, who also works as an applications scientist at Agilent, will discuss data interpretation of N-glycan profiles. And Dr. Aled Jones holds the position of technical liaison at Prozyme. Aled is going to provide an overview of the GlycoPrep Plus platform for high-throughput N-glycan sample preparation. After the panelists have made their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So let's get our webinar up and running. I'd like to introduce our first speaker, Dr. Oscar Potter. Oscar?
Thank you very much. In this part of the presentation, I will give a brief background on N-glycan analysis by hydrophilic interaction chromatography. My colleagues will then follow with more detailed information on sample preparation and analysis. N-glycosylation is a common post-translational modification with potential N-glycosylation sites found on more than 50% of human proteins. Many therapeutic proteins contain N-glycans, including IgG-type monoclonal antibodies, which have a conserved N-glycosylation site at asparagine 297 in the Fc region of the heavy chains. The presence and structure of the N-glycan at this position is critical to some of the biological functions of IgG. Some glycan structures are important for efficacy, while other structures can have consequences for safety, clearance, and immunogenicity. N-glycosylation is therefore a critical quality attribute in monoclonal antibody development and production, and a thorough understanding of glycosylation can furthermore be helpful for IP reasons or for the purpose of demonstrating comparability for a biosimilar product. N-glycans are a very complex type of post-translational modification because they are composed of several types of monosaccharide, which are connected in a branched structure by various types of glycosidic bond. All glycan structures contain the core structure, shown here in CFG and Oxford-style notation. The range of glycan structures which can be built on top of this core is dependent on the biological context. For the biopharma industry, this context is usually humanized cell lines from one of a handful of popular cell types. In the case of monoclonal antibodies based on IgG, we expect to see glycans from just two broad families. Most of these will typically be complex-type glycans with one or two antennae, and many will have the core fucose group attached, as shown here by the red triangle. We may also see some high mannose-type glycans, which contain only branched mannose structures in addition to the glycan core. N-glycan analysis can be performed at three levels. The first level is the analysis of the intact protein by mass spectrometry, typically by LCMS using a short reversed phase column hyphenated to a high-resolution time-of-flight mass spectrometer. Deconvolution of the mass spectrum provides the intact mass of the antibody, including the two N-glycans which are typically present. The analyst can determine the mass that was contributed by the two N-glycans by repeating the analysis on the sample of the antibody which has been deglycosylated by PNGase F. Whilst the total mass of the N-glycans is not usually enough information to assign glycan structures with high confidence, many analysts appreciate the speed and simplicity of this approach. Some analysts also use an enzyme such as IDES to digest the antibody into subunits that contain only 1/2 of the Fc region, and therefore only one N-glycan. The second level of analysis is to determine the N-glycans at the glycopeptide level as part of the peptide map. Whilst this approach offers only limited information about glycan structure and relative quantity, it is nevertheless an attractive approach for analysts who are already running peptide maps in order to confirm protein sequence and other post-translational modifications. Finally, many analysts choose to analyze glycans after they have been released from the protein by an enzyme such as PNGSF. This approach offers the most accurate information about structures of the glycans present and their relative quantities. Today, we will talk about analyzing released glycans by HILIC with fluorescence detection. However, it should be pointed out that there are other types of separation which can be used to study released glycans, including capillary electrophoresis and other types of chromatography. In addition to the methods mentioned above, Agilent recently introduced an all-in-one solution based on microfluidic chip technology and mass spectrometry. The reusable MAB glycochip performs online enzymatic release of glycans, as well as sample cleanup, separation, and nanoelectrospray ionization. Coupled with a time-of-flight mass spectrometer, the MAB glycochip solution can be used to generate a glycosylation report from a pure monoclonal antibody sample in as little as 15 minutes. Amidst the various options of N-glycosylation analysis, HILIC separation of released fluorescently labeled glycans has become the method of choice in many laboratories. When glycans are released from a protein and labeled with the fluorescent tag, each glycan molecule will generate a strong and equal signal as it passes through an online fluorescence detector. This means that a HILIC fluorescence chromatogram allows true relative quantitation without the need for any calibration. Since the glycans are no longer attached to the peptides or proteins, they can be well separated based on their structure, and, if desired, they can be also interrogated in a mass spectrometer without the confounding influence of the peptide or protein backbone. Labeling also causes each glycan to elute as a single sharp peak, whereas unlabeled glycans would produce a broad or split peak due to anemorization of the monosaccharide at the free reducing end. HILIC with fluorescent detection is a robust method with high convenience. The laboratory experience is nearly identical to performing reversed phase chromatography with a UV detector, so training is relatively easy and many labs will already have most of the instruments that are required. There are several ways to interpret a HILIC fluorescence chromatogram of N-glycans. At a basic level, the chromatogram provides a relative profile of the N-glycosylation that can be used to monitor consistency between samples. More often, the analyst will assign the peaks to particular glycan structures. The data can then be tabulated and simplified into categories so that we can report properties such as the relative amount of high mannose glycans compared to complex glycans, or the relative amount of fucosylated versus non-fucosylated structures. Agilent has recently released 2 HILIC columns for glycan analysis. The first is a column that uses superficially porous particles with a diameter of 2.7 microns. The column is recommended for customers who want to work at lower pressure, for example, customers who do not want to upgrade their HPLC equipment. The second column uses sub-2 micron-sized particles for ultra-fast separations on instruments that are capable of operating at ultra-high pressure. HILIC analysis of fluorescent-labeled N-glycans is a robust method for in-depth analysis of therapeutic protein glycosylation. However, new users will discover that there are a few challenges. At the end of the workflow, accurate peak assignment can sometimes be difficult, even for experienced users. There are a variety of tools that can facilitate peak assignment, including retention time standards and enzymes for glycan sequencing. Later in the presentation, you will see an example of peak assignment by online mass spectrometry. At the start of the workflow, the sample preparation procedure involves multiple steps and several different consumables. I will now hand the presentation over to my colleague Jim Marticella so that he can explain the N-glycan sample preparation.
Good afternoon. In this section of the webinar, I'd like to discuss the sample prep steps and methods involved for release and labeling of glycans for analysis by HPLC and mass spectrometry. However, before we get started, I'd like to review some key terminology. When we talk about the majority of glycan profiling of therapeutics, such as monoclonal antibodies in this example, we are referring to N-linked glycans. In general, the linkage of carbohydrates to proteins can occur through either O linkages or N linkages. O-link glycans attach to the oxygen atom of the side chain, or serine, or threonine. However, the more common form of glycosylation is N-link glycosylation, where the attachment of the glycan is on the amide nitrogen on the side chain of asparagine. This nitrogen attachment, you can see here by the blue arrow, is called the N-glycosidic bond. This bond is between the glycan, beginning with an acetylglucosamine sugar, or referred to as glucanac, and the monoclonal antibody. Its relative importance in this talk is to point out the site of enzymatic cleavage during glycan sample prep. Free-released N-link glycans are undetected by UV detection and are commonly labeled with fluorescent dyes or tags at their reducing ends. Labeling with fluorescent tags increases absorption by fluorescent detection and provides enhanced and sensitive signal response for efficient glycan profiling. Categorized in this slide are the three most common fluorescent tags used for N-link glycan profiling. 2AB, or 2-aminobenzamide, is the most popular label and delivers excellent separation performance with high sensitivity by fluorescent detection. 2AA, or 2-aminobenzoic acid, is also a common tag, but better suited for negative mode MS analysis. And finally, procainamide provides superior sensitivity in MS-positive mode and is becoming the more popular choice for MS analysis of N-link glycans. So let's talk about the sample preparation workflow for N-glycan profiling. Releasing and labeling glycans from glycoproteins, such as monoclonal antibodies in this example on the slide, are really two distinct processes: deglycosylation, or glycan release, and labeling the released glycan. The first step, deglycosylation, is shown here in this cartoon, and it involves enzymatic cleavage of the glycan from the antibody followed by a cleanup step. Breaking this down a bit more, though, the first part of the deglycosylation results in a mixture of the released N-link glycans, the free antibody, and the enzyme and reagents used for deglycosylation. Moving over a little further on this slide, in the second part of this workflow, the released glycans need isolation from the antibody and reagents for the downstream labeling step. The purification of the glycans then require a solid phase cleanup step, which is most commonly performed by HILIC separation. We can take a look at these two processes a bit more closely. Here's a look at the deglycosylation chemistry and what's important for the efficient release of N-link glycans from glycoproteins. Let's start with the enzyme, peptide N-glycosidase F, or more commonly referred to as PNG-ACE-F. This enzyme is the most effective enzymatic method for removing almost all N-link glycans from glycoproteins. It works by cleaving between the innermost glucanac and asparagine. PNG-ACE-F digestion deaminates the asparagine and leaves the glycan intact. This keeps things manageable then for the fluorescent tagging steps that are to follow. However, it is important to point out here that there are physical factors that can inhibit the completion of this process, and that denaturation and elevated temperature can greatly increase the rate of deglycosylation. Heating to 37 degrees C and using denaturants such as SDS and DTT are recommended for complete deglycosylation. It is also important to mention that there are other enzymes available, such as endoglycosidase H, but most are not suitable for general deglycosylation of N-link sugars because of their limited specificities, and also because they leave one N-acetylglucosamine residue attached to the asparagine. After deglycosylation, N-glycan sample isolation is achieved by solid phase extraction on a HILIC sorbent, as I previously described. Taking a more detailed look here at this cleanup process, the released glycans and free antibody can be processed through a HILIC sorbent that will bind the free N-glycans, while the deglycosylated antibody and residual enzyme reagents are washed away through with an organic rinse. As a reminder, in HILIC separations, the polar glycans will stick to the HILIC sorbent under organic conditions, while the organic soluble materials are free to pass through into the waste stream. This process is shown in step 1 above to the left. In step 2 to the right, the glycans bound by the HILIC sorbent can be isolated for labeling by the addition of subsequent water washes. The water washes release the glycans for collection, and because no polar reagents were used during deglycosylation, they are now very clean for the fluorescence labeling steps. The second part of sample preparation for N-glycan profiling is fluorescence labeling of the isolated glycans from D-glycosylation, followed again by solid phase cartridge cleaning steps. You'll see, too, that these steps are nearly similar to the D-glycosylation cleaning steps. First part of fluorescent labeling or tagging involves a chemical reaction between the glycans and the fluorescence tag. After tagging, the glycans, the residual dye, and the reaction components from the labeling reaction need to be removed. Efficient removal of these components away from the labeled glycans will ensure that you get clean HILIC chromatography and provide high-efficiency separations and improved retention without interfering background effects. As shown in the previous slide with the D-glycosylation breakdown, I'll do the same with the labeling chemistry and cleanup. In the interest of being technically accurate when the glycan is first released after D-glycosylation, I wanted to highlight a simple conversion shown in the top panel above. In this view, when the free glycans are enzymatically released from the protein, they are in the glycosylamine form, as shown in the upper left. However, for the labeling reaction to proceed, this free amine glycan must be converted to a hydroxyl so it has a free reducing end for the labeling reaction. This is a simple conversion under acidic conditions. Looking at the labeling chemistry in the bottom panel, glycans are labeled in a two-stage reaction. First, the free reduced N-glycan is in its open ring structure conformation and reacts with the fluorescent tag to form a stable Schiff's base. This Schiff's base is then readily reduced to yield a stable labeled glycan as shown in the bottom right. In this example, with 2AB as the tag, the labeled glycan would then result in a 120 Dalton mass addition. It is important to note that glycan labeling is a very robust and stable reaction with no degradation components. Additionally, it does not need a post-cleanup analysis for analyzing chemical byproducts from this reaction. However, in these reactions, there is commonly a large excess of 2AB dye reagent, and robust sample cleanup is needed to free the labeled glycan of both this residual dye as well as the matrix components, such as the solvents and the reductant. After labeling, and shown here, the post-labeling cleanup is similar to D-glycosylation cleanup, as you will find. Labeled N-glycan sample purification is again achieved by solid phase extraction on a HILIC sorbent as previously described. Reviewing this process and taking a more detailed look specifically for labeling cleanup, we see the crude to-be-clean labeled glycan mix contains excess dye and matrix. This mix is first passed through a HILIC sorbent that will bind the labeled N-glycans while the dye and matrix are removed with an organic wash. Again, in HILIC separations, the labeled glycans stick to the HILIC sorbent while the organic solubles pass through into the waste stream. Then moving over to the next step, step 2 to the right, the HILIC-bound labeled glycans are eluted by the addition of water washes. The water washes release the labeled glycans from the HILIC sorbent, and the labeled glycans are then collected. Commonly, the collected labeled glycans are dried down and resolubilized in an appropriate injection solvent for HPLC, FLD, or mass spec analysis. In summary, and shown under the description heading, D-glycosylation involves enzymatic treatment of the glycoprotein followed by HILIC cleanup in a first step and labeling with a fluorescent tag or dye followed by HILIC cleanup again in the second step. In the right-hand section of this chart, you will see I provided Agilent D-glycosylation and labeling kits that can help achieve a successful labeling procedure for your analysis. The two kits are offered in low sample and large sample options and are currently available to accommodate the protocols described in this talk today. I would now like to point your attention to Sonia Schneider, who is going to discuss the data interpretation portion of a labeled glycan analysis.
Thanks, Jim. So welcome to my part of the talk. Today, I'm going to talk about data interpretation after fluorescence detection and online MS detection. You see here the separation of N-linked glycans from a rat monoclonal antibody. The glycan pattern was optimally resolved and allowed the separation of all major N-glycans. Some customers are already satisfied with the FLD chromatogram because they only need to compare the glycan profile of different production lots of their biopharmaceuticals. In this case, they are especially interested in relative quantification of the single glycans after peak integration. After integration, the peak area percentage was calculated for relative quantification. The results are shown in this table to the right. High intensity of the labeled glycans was achieved by setting the optimal wavelengths for glycan detection on the Agilent 1260 Infinity fluorescence detector using 260 nanometers as excitation wavelengths and 430 nanometers as emission wavelengths. However, some customers may need to assign different glycan structures for their purposes. There are various approaches to identify the peaks, and I will now show you how to do it using online MS with a QTOF. As an example, let's have a look at the spectrum at the corresponding elution time for peak 5. A strong signal could be observed at 873.34. As glycans ionize typically as AIM plus 2H ions in positive electrospray mode, the detected glycan has a mass of about 1,745.68. This mass is then used for further investigation. A free online database was used to find related glycan structures of the detected parent mass. The found mass was entered into the glycamod tool from efficacy. In this tool, the experimental masses are entered in combination with important further parameters needed for identification. Further parameters are the found ion mode, the form of the glycans, how they are present in the sample. In our case, we are looking at derivatized glycans. In this context, it is also important to specify the used derivative. Here, the used labeling reagent 2AB and the corresponding mass of 136. Having entered this information, the database proposes 11 glycan structures that could potentially represent the search glycan. In the case of 873, this slide shows you 3 structures examples that are theoretically possible for this mass. The glycan could either be core fucosylated, like shown here on the right and below. However, outer arm fucosylation is also possible for this mass seen on top. The glycan below contains, for example, one more mannose instead of galactose and GalNAc instead of glucnac. How is it possible that all 3 masses are isobaric masses? The mass of the glycan results from the monosaccharide composition, which are shown here. Several monosaccharides have the same monoisotopic mass, like for example seen for galactose, mannose, or glucose, and also for glucnac and GalNAc. This fact leads to several glycan structure possibilities that could have the same mass. During fragmentation in MS-MS analyses, the sugar chains are cleaved between the monosaccharides. Are there any rules for typical cleavage sites? Here we see the typical fragmentation of N-glycans. The glycans tend to fragment between glucnacs and mannose units, like it is shown here with the red arrows. In addition, sialic acids are usually cleaved as well. Targeted MS-MS data was then used to distinguish between the potential structures suggested by the glycamod tool from efficacy. Here you see the CID MS-MS spectrum of the N-glycan with the mass of 873. The strong signal of 204 represents glucnac. With the addition of 262 Dalton, an attached hexose, for example galactose or mannose, are found. Because glucnac and mannose 5 are usually cleaved during fragmentation, we would expect a mass of 407 representing glucnac and GalNAc from the first glycan. As there is no peak 407 found and the mass 893 represents the 3 mannoses together with 2 attached glucnacs, the first proposed structure becomes very unlikely. so 2 more structures are left. Outer arm fucoses are usually represented by a mass of 212 or 350. Therefore, due to the fact that these masses are missing in the spectrum, there is a higher possibility for a core fucosylation. The core fucosylated version is also supported by the masses of 1,177 and 1,542. For another detected glycan mass, 2 possible structures are proposed by efficacy containing different sialic acids. To identify the correct one, the MS-MS spectrum of this mass is analyzed in detail. The MS-MS spectrum shows a strong fragment ion signal at 308, representing mu GC, and 673, representing mu GC attached to galactose and glucanac. The presence of those 2 strong signals and the missing of 292 or 657 that would have indicated the presence of mu AC confirms the glycan containing mu GC as sialic acid. On this slide, you can see the results of the glycan assignment of the rat monoclonal antibody that were all assigned in the same way. Regarding the nomenclature, this table shows the Oxford style where the second peak is named FA2. In comparison, the biopharma map style uses GOF for the same glycan. In addition to the nomenclature, the detected mass, the peak area percentage for relative quantification, and the drawn structure is shown. With this table, it is now possible to answer the typical biopharma relevant questions like already mentioned from Oscar in the beginning. These answers are also summarized in this table. After identification and relative quantification, we are able to compare the amounts of neutral glycans versus charged glycans, hymenose versus complex glycans, and fucosylated versus non-fucosylated glycans. In this rat monoclonal antibody, 71.3% of the glycans were neutral and 28.7% charged, having mu GC as sialic acid attached. Only mannose 5 was detected as hymenose glycan and was also the only one found that was not fucosylated, so we see here 1.2% for hymenose and for non-fucosylated glycans. This slide shows the separation of ovalbumin glycans. Ovalbumin is N-glycosylated only at one site, but it has a complex glycosylation pattern. The high performance of the glycan mapping column allowed resolution of over 50 peaks with a good signal-to-noise ratio. 20 major peaks were assigned based on the parent ion mass data. Detailed structural conclusions were not achievable due to the high chance of isobaric structures occurring, several of which cannot necessarily be distinguished from the MS-MS data. Here we see an example of an isobaric structure where, for example, the galactose could be attached to 4 different glucanacs. This makes a unique identification impossible for MS, sometimes even for MS-MS data. Instead, the N-glycans are described in terms of their monosaccharide composition, like it is seen on this table, showing the mass plus 2AB, the calculated mass without label, a short form from the composition with H for hexosis and N for glucanac. I would like to thank you for your attention, and I pass on to Alec Jones from Prozyme, who is going to talk about automated high-throughput methods for sample preparation.
Thanks, Sonia. In the last segment of this webinar, I'd like to discuss Prozyme's GlycoPrep, a sample preparation platform for N-glycans as automated on the Agilent AssayMap Bravo liquid handler. I'd like to give a brief overview of this N-glycan sample preparation system, and then show you some qualification data using a number of different glycoproteins, so Rituxan, a human IgG and RNase B mixture, and also N-brel. So this slide gives a summary of GlycoPrep. It's a modular sample preparation platform for N-glycans. Up front, there's an optional protein A purification module to purify antibodies from conditioned media. And then you take your purified glycoprotein and denature it and immobilize it on a reverse phase matrix. You then digest with N-glycanase or PNGase F to release N-glycans. These released N-glycans are then fluorescently labeled, and as Jim has touched upon earlier in this webinar, the excess dye is then cleaned up with a HILIC resin, and the eluted N-glycans in water can then be used for analysis. It's a modular design, so you can switch out labels. It's quantitative and can be performed within 2 to 5 hours, which is quicker than the traditional glycan preparation methods, which sometimes take days. These are the labeling chemistries that are available with this platform. I'm going to focus on 2AB today, as others have, but I'd like to point out that other labels are available depending on the analysis. APTS can be used for capillary electrophoresis, and instant dyes can be used for faster labeling. Also, we have a procainamide label in development for sensitive MS. Glycoprep N-glycan preparation can be run on a number of different formats, 3 formats, 2 of them manual. The first is in a microfuge. This is for up to 24 samples, and this can be run with microfuge tubes. The second is a manual format using microplates for higher numbers of samples. And the final one, which I'll focus on today, is the automated format. This, again, uses 96-well microplates and uses the Agilent AssayMap Bravo. The technology is really focused on the use of these AssayMap cartridges, which contain a 5 microliter packed resin bed with an upper and lower frit. So it's true microchromatography as opposed to a packed pipette tip. These interact very nicely with the Agilent AssayMap Bravo liquid handling workstation. The probes form a tight seal above the upper frit so that samples and reagents can be aspirated and dispensed through the matrix. So in summary, we've really taken a traditional workflow with traditional chemistry and optimized the conditions and streamlined it and speeded everything up. You can use up to 50 micrograms of any glycoprotein per preparation. Our current commercialized product is the 2AB workflow on the Agilent Bravo. It takes around 4 to 5 hours per plate, and the output is labeled N-glycans in water. So I'm just going to give you a few examples of data obtained using the system. The first is with 4 lots of rituximab, 24 replicates each, and analysis is by UHPLC using a HILIC 10-minute screening method. We inject around 20 nanograms of N-glycan sample in aqueous buffer. Here's an example of a chromatogram showing HILIC separation of N-glycans from rituximab, and you can see here the major peaks such as G0F glycan and G1F. And here's the data from those different lots plotted by glycan with a relative area percentage on the y-axis. You can see that within the lots, the data is pretty tight. The error bars are very small. But between lots, there are some differences between the levels of each of the different glycans, such as G1F and G0F. And this is what people look out for. Here's the data plotted by table format. Across the top here, you see the 4 different lots of Rituxan. And highlighted in red are the percentage CVs. And these are pretty low. So, for example, for G0F, you can see 0.5%, 0.3%. And really the take-home message from this slide is that percentage CV for peaks greater than or equal to 1% abundance is less than or equal to 2.3%. The second example I'd like to show you that uses this N-glycan sample preparation system is using a glycoprotein mixture. So this is human IgG mixed with bovine RNase B, and this is 48 replicates. And we use this mixture because it contains species that you would see in monoclonal antibodies, such as G0F, but also we use RNase B because it contains high mannose N-glycan species. Again, we analyze the data using HILIC, and this time, because there are a lot of glycans present in the sample, we use a 60-minute gradient for a greater resolution. Here's the data shown as overlays of all 48 replicates, and again, the major peaks such as G0F and G1F. Here's the data for human IgG and RNase B N-glycans, 48 replicates in table format, and you can see that for the major glycans, again, such as G0F, percentage CV is quite low. And this slide shows the results across formats. So manual formats, such as using the microfuge format or the manual 96-well plate format or the automated 96-well format. And this is to show that we get good agreement between each of these methods. So really people can pick the method that they would like to use. The final example I'd like to show is using N-brel, 15 replicates, again using the 2AB workflow on the Agilent assay map Bravo, and a 60-minute HILIC separation. These are the separations from the 15 replicas presented as an overlay, and again the major peaks such as G0F and G1F. Also in this sample you see silylated glycans such as A1, A1F, A2, and A2F over on the right-hand side of the HILIC FLD chromatogram. The data is table format. Again, G0F, percentage CV is 0.8, and the percentage CV for peaks greater than or equal to 1% abundance is less than or equal to 2%. So in summary, this is a high-throughput N-glycan sample preparation system that can be automated on the Agilent assay map Bravo. You get an unbiased N-glycan profile. It supports orthogonal and hyphenated methods such as LC, CE, and LCMS. It's available in multiple processing formats such as microfuge, centrifuge, partial and full automation. It's reproducible as shown by the percentage CV data, and it's transferable. All kit components come from Prozyme, so there's no need to source from multiple suppliers. I'd like to acknowledge my colleagues who contributed to this work. This concludes my portion of the webinar, so I'd like to hand back to John at this point.
Thank you, Alid, and thanks as well to Oscar, James, and Sonia for your excellent presentations. You've given our audience some wonderful tips, insights, and advice on how to better achieve successful N-glycan analysis. Thank you. As a reminder to our audience, feel free to send in questions for any of our panelists. Just type it into the Ask a Question box on your screen and then hit Submit. We're almost ready to begin our Q&A session, but first, I'd like to ask you to disable your pop-up blockers because a short survey on this webinar will be appearing in a moment. We would very much appreciate your feedback. Okay, let's begin the Q&A, and we'll take a look at the first question from our audience.
Oscar, first question is for you. Can we use those HILIC columns in reagent TAC for determination of alpha and beta glucans, not glycoproteins?
Right, so with the Agilent's new HILIC columns are an amide type HILIC column, and this type of column is appropriate for separating various other types of carbohydrate in addition to N-glycans. So for example, you can separate dextran or other small carbohydrates or medium-sized carbohydrates like molapectin or heparins, glycosaminoglycans as well.
And we have another question, Oscar. Is there any way to analyze glycans without mass spec?
Yes, there are a few different approaches. So one is to do the assignment based on retention time. And the simplest way to do this is to purchase a standard. So if you think that the glycan is, for example, the G0F, you can purchase that glycan from a company like Prozyme and do an injection and see if the retention time matches up. Another popular approach is to use exoglycosidases, which selectively cleave a monosaccharide with a particular linkage from the glycan, and by doing a sequence of digestions you can sort of strip the glycan back to do a sort of reverse construction of the glycan, and that can help you assign it as well.
Okay, and Oscar, we have another one here. According to your experience between CE and LC, which technique is more appropriate for glycan analysis characterization in terms of selectivity, MS coupling?
Right, so with CE, there's two main ways that that's done. One is CE with laser-induced fluorescence detection without coupling to mass spec, and that is a very high resolution approach with sort of orthogonal selectivity compared to HILIC. Some people like that method, that they sort of prefer that because they like the high resolution. Modern HILIC with these new columns is nearly as good in terms of resolution. However, some people prefer it because of the ease of use or just being more familiar with HPLC. In terms of MS coupling, you can do CE-MS as well as just as you can do the HILIC LC-MS. However, that's a little bit more complicated, and I think that overall it's easier to couple the HILIC with mass spectrometry than it is with the CE, although some people have had success with the CE-MS coupling.
Thank you for that, Oscar. Jim, question for you. How much laboratory time does it take to do the hands-on manual D-glycosylation and labeling process as shown in the presentation?
These times can really vary, and they have a number of dependencies. I think one of the main dependencies is how familiar you are with laboratory equipment and sample processing for SPE and these sorts of things. A low number of samples, somewhere around, say, 96, can on average be completed in about 2 days from both release to labeling. However, if you're looking at larger sample quantities to process, I would have the expectations would be for extended periods of times, perhaps upwards of 4 days.
Okay, and related question, the D-glycosylation and labeling process looks a bit complicated with many steps. Can this be done by users with little or no background in glycan sample prep?
This really depends on how the reagents and supplies are purchased. With little background, I think you might be more inclined to purchase sort of all-in-one sort of protocols or packages. This would make things a lot easier. Going about procuring all these independently can be a bit time-consuming and challenging, trying to navigate through the different reagents and suppliers.
Okay, thank you. Sonia, I have a question for you. Can online mass spectrometry be used to distinguish between glycan linkage isomers, for example, 2,3 versus 2,6 linked sialic acids?
Well, the type of online mass spectrometry that we discussed cannot distinguish between those linkage isomers. Some expert glycans, while some expert glycan mass spectrometry methods can distinguish between some types of linkage isomers, it is usually easier to assign linkage isomers based on retention time or using exoglycosidases like Oskar already explained. In practice, many analysts working with monoclonal antibodies usually avoid these steps by assuming that the glycans have the most common types of linkage.
Okay, Sonia, and we have another question. Why not use negative mode? This should provide better MS-MS spectra? Because that's a question.
Negative mode can be used for better structural information, but when you have 2AB labeled glycans, sometimes it cannot be sensitive enough, especially when you don't have a lot of sample.
Okay, well, thank you. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you miss parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. Thank you again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your questions about the various topics that were brought up during the webinar. And thank you to Agilent, whose support made this webinar possible. Bye for now.